Hello students, today we'll run a practical on study of structure of prokaryotic and eukaryotic cells. In introduction, cells are the fundamental units of all living organisms ranging from unicellular prokaryotes to multicellular organisms. In order to define cell, the cell theory was proposed around the 19th century, which states that every living organism is constituted by one or more cells, that the cells are the functional unit of all living organisms, and all cells arise from pre-existing cells. Though cells of microbes, plants, and animals differ in shape, they share a lot of common features, such as having a plasma membrane to limit the cellular content, cytoplasm that serves as a matrix for all life activities, information coding DNA and RNA, or ribosomes for protein synthesis, just to name a few. Broadly, cells can be classified into two categories based on the location of their genetic material. The prokaryotes, pro meaning primitive and karyon meaning nucleus, and the eukaryotes, in which eu meaning new and karyon meaning nucleus. The prokaryotic cells do not have a definitive nucleus, whereas the eukaryotes have true nucleus in which the genetic material is enclosed, though both types of cells have DNA as genetic material. Comparatively, prokaryotes are much smaller in size, ranging from 0.1 to 5 microns, whereas eukaryotes has a size ranging from 10 to 100 microns. Prokaryotes do not possess specialized organelles as seen in eukaryotic cells. Now let's discuss more about eukaryotic cells. All eukaryotic cells originate from a common ancestor and shares many common features. One of the organelles of eukaryotic cell, the mitochondria, is thought to originate from an endosymbiotic event that formed a stable coexistence. Mitochondria is the energy house of a cell and is responsible for generating ATPs, the energy currency of a cell. The organelles has its own genome and is similar to prokaryotes. The smooth and rough endoplasmic reticulum, Golgi apparatus, ATS-type ribosomes, vacuoles, and lysosomes are other common organelles that are present in almost all eukaryotic cells. Rough endoplasmic reticulum, present close to the nucleus, and its rough appearance is due to the presence of attached ribosomes. On the other hand, smooth endoplasmic reticulum are sites of secretion of lipid-based molecules and also functions as detoxification centers of harmful chemicals. This is why the liver cell contains abundant smooth endoplasmic reticulum. In muscle cells, smooth endoplasmic reticulum acts as calcium reserves and helps in muscle contraction. Golgi apparatus functions as center for sorting, modification, and packaging of cellular products into vesicles such as lysosomes. Lysosomes act as sites of digestion of food particles that are endocytosed by cells. Vacuoles functions as site for water storage within cells that regulate the water content and store metabolic products or poisonous molecules. Specialized organelles of eukaryotic cells. Eukaryotic cells also has distinct cell organelles that are specific for each kingdom. As for example, kingdom Plantae and Animalia, which are both eukaryotes, have specialized organelles. Unlike animal cells, plant cells have chloroplasts that are required for photosynthesis. Moreover, they have cell wall, which is a rigid layer made of cellulose that supports growth and also helps in water retention. Plant cells have large vacuoles to maintain water pressure in the cell. Additionally, plant cells have plastids that are meant for storage of products of photosynthesis. In terms of shape, plant cells are rigid and have almost rectangular shapes, whereas animal cells are more irregular and are rounded. Microscopy in studying cells. Cells differ in size and shape. Some cells, such as frog oocytes, are large enough to be observed with naked eye, and yet most cells are not visible to the naked eye and needs visual aid such as microscope. Though microscopy is a powerful tool, most tissue lack pigmentation and need special dyes to visualize under a microscope. Therefore, solutions that can specifically stain molecules and organelles according to their chemical nature has been created. Some of the examples of such stains are methylene blue that stains nucleic acid blue by binding to negatively charged DNA, safranin that stains nuclei red or pink. After staining for a brief period with the dye, cells are mounted on a glass slide, wet or dry. A wet mount is created by placing the sample on a glass slide with liquid between the slide and the cover slip. The liquid used may be glycerol or other liquids like water. To further enhance the magnification, oil can be used on top of the cover slip when using high magnification. Materials required for the experiment. We will be needing bacterial culture, plant or animal cell, a bright field microscope with 40x and 100x objectives, glass slides, cover slip, and immersion oil. In the procedure, preparation of animal cell slides. First, pour around 30 ml of double distilled water into a beaker. Using a glass dropper, place 2 to 3 drops of water on the center of a glass slide. With a clean toothpick, scrape the inside of your cheek to gather cells from the buccal cavity. Care should be taken not to injure the inner wall of your cheek. Place the cheek cells from the toothpick on the wetted area of the slide and mix gently. Now, add 2 to 3 drops of methylene blue stain solution on the wetted area of the slide, followed by mixing. Let it stand for around 3 minutes. After 3 minutes, place a drop of glycerol using a glass dropper. Now, place a cover slip by letting stand one end of the cover slip and gradually lowering the other end, making sure there are no air bubbles between the slide and the cover slip. Now, remove excess liquid by using a blotting paper. Preparation of bacterial cell slides. As bacterial cells lack definitive nucleus and are small, a light microscope is not good enough to study detailed structure of the cell. However, certain staining methods can enhance the contrast to identify morphological features of a bacterial cell. For preparation of bacterial cell slide for microscopy, approximately 100 microliter of curd is mixed with 500 microliter of distilled water. In the next step, a drop of the mixture is taken and placed on a clean empty glass slide, followed by preparation of a smear using another glass slide. Now, dry the slide over a flame. Make sure that the slide is not overheated. A drop of methylene blue is added over the smear and kept for 3 minutes. Now, excess stain is removed by dipping the slide in a coupling jar containing water. Next, remove excess water from the slide and place a cover slip and observe the slide under a microscope after covering with a cover slip. In the results, methylene blue stains negative charge parts of a cell. In case of eukaryotes, a clearly stained nucleus can be observed along with a translucent cytoplasm within a boundary of the cell membrane. In prokaryotes, no definitive nucleus can be observed, but the shape of the cells, which are rod-shaped in case of lactobacillus from yogurt, are observed stained with blue. In conclusion, prokaryotic and eukaryotic cells differ in shape and complexity. Eukaryotic cells are larger than prokaryotic cells and contain internal organelles that are meant for specific functions. Since cells are larger, eukaryotic cells and organelles can be individually visualized by using specific dyes such as methylene blue that stains the nucleus. However, this procedure is not possible for prokaryotes as they lack specific nucleus and are smaller in size. The experiment that we have just learned enables us to distinguish prokaryotic and eukaryotic cells under a light microscope. Thank you.